So good morning, everybody, and welcome to the 2017 RSN conference. Just before we start, just a few housekeeping things. So there are no scheduled fire alarm tests today, so if you do hear the fire alarm, the exits are through that doors there. That's the easiest way to get out of the building, and there'll be wardens who will chaperone you out of the building. Toilets, there's a multi-access one on this level. Ladies is slightly downstairs if you go out the doors and down. Gentlemen is one up. Don't go through the first door like I did, because that's the broom cupboard. So there's also some stands outside, so please visit those during the breaks as well. So we've got information from the local development team there. We've got a book of poetry. So one of the RSN volunteers, Anna, has wrote that with your mother, wasn't it? They're your mother's poems, that's right, okay. And also we've got information from one of our speakers' projects today, which is on the tables as well. Okay, and before I also start my own presentation, I'd like to take the time to actually thank Anna and a fabulous team. Through her dedication and hard work, has really driven to make the RSN such a successful network. Also, I'd like to thank the volunteers that have come from Parkinson's UK today to help the day run smoothly. And also, I'd like to specifically thank James, who's done such hard work in the organisation, and I'm sure that we'll have a fantastic day due to his hard work with the organisation. So without further ado, I'll just introduce myself because, as you know, I've recently joined Parkinson's UK as of the 1st of March. So many of you may recognize my face because I was sort of the public face of the tissue bank at Parkinson's UK as well. So I've come from Imperial College and I'm in charge of what's called scientific programs. So I'm in charge of the university grant schemes, so I'm managing a budget of about 4.3 million a year at the moment. So this covers project grants, fellowships, things like the Oxford Discovery, the Monument Foundation Award there, the cohort studies as well. So I also manage the Cohort Studies Council, and so these oversee our cohort studies, and they also assess applications for future cohorts. Also, I manage the critical path for Parkinson's, and this is really starting to pay benefits now because we're really starting to radically reshape our clinical trials so that when we drive drugs through the development pipeline, that we've got clinical trials which are fit for their testing. So I'm also involved in the drug discovery side with the virtual biotech. Now, as you know, that this was launched earlier on this year. And the reason why we've launched the virtual biotech is really to try and drive through the drug discovery program. Because with our traditional funding, academics would get their ideas about a drug to this point. And at that point, they would need a massive injection of cash, generally from pharmaceutical companies, to take it into the clinical trials. Now, as you know, there's lots of failures in CNS drug development, particularly in the Alzheimer's field recently. And these studies cost millions. And so pharmaceutical companies are reluctant to come to the table to take on these projects. So what we're doing with the virtual biotech is actually bridging this gap. So rather than having the potential drugs which could be treatments dying in this valley of death here, we're giving the bridging funds which will allow the further development of these compounds to a point where they're more attractive to the pharmaceutical companies. So just a little bit about my background. So I was the professor of neuropharmacology, so it's all about drugs and diseases, really. And I was the deputy head of division of brain sciences at Imperial College. So we had about 67 academics within the division there. I'll say before I was the director of the Parkinson's UK Tissue Bank, and I established it there in 2002. So all in all, I had 32 years of research in the Parkinson's field, and I've had a long association with Parkinson's UK. Indeed, at the beginning of my career, they actually funded my PhD. So 23 years of this was based at Imperial College. And my main areas of research were into trying to find out what was causing the cells to die in Parkinson's disease and developing new drug therapies, which we tested through preclinical stages and even got them to the clinic. So some of the things that some people might know me for is with Tony Shapiro's group, we were the first people to show that there was a defect in the mitochondria in Parkinson's disease, and that there is evidence of these nasty free radicals damaging the tissues within Parkinson's as well. So also, my researchers and myself were the first people to show that there was a conclusive increase in iron within the Parkinson's Niger, and we tested iron chelators within the clinic. So just looking about our strategy and how we're doing, what we're saying is we need to be international leaders, and we were catalyzing 2 projects as our aims. We've, as you know, the Critical Path for Parkinson's and the Innovations Medicines Initiative is 2 projects that we've already launched. Also, what we wanted to do is have 2 to 4 drugs in the pipeline and 2 to 4 drugs in the preclinical phases. GDNF trial is already completed, and later on this year it should report, and this might lead to a phase 3 trial. So Virtual Biotech 1 has been launched, which is Keepstone, which is a project jointly with Sheffield University. The Virtual Biotech 2 has just been announced, and this will be a novel treatment for L-DOPA-induced dyskinesias. So we're rapidly accelerating through the release of the Virtual Biotech program, and Virtual Biotech 3 may be possibly released before the end of the year. Also in 2018, we'll hopefully do some repositioning drug trials as well. So we're well on target for meeting quite a lot of our aims in our strategy already. So we're also analyzing the way that we fund research through what's called Research Fish Data, and that is being presented to the Research Strategy Board later on this year. Effective communication, our communications team is actually doing a fantastic job. We've got several publications, we're on the web, we do media releases, blogs, Twitter, so we try and cover every base about trying to get information to you. So patient involvement, we're leading the way. If you speak to the AMRC, which is the overarching things for medical charities, we are the envy of these charities as well with our patient involvement. And what we do is involve patients at every level, and this really leads me into the introduction of the RSN. Thank you very much.
Thank you very much, David. So I'm now going to hand over to Simon Cran-McGreehin, who is the chair of our Research Support Network Development Team, and he's going to talk to you about what they do and a project that they've been working on. Thank you.
Okay, good morning, everyone. If I press this a couple of times, there we go, excellent. So, my name's Simon, and I chair the development team on the Research Support Network. And the development team is a group of about 10 or 12 volunteers and staff, and the volunteers are drawn from the Research Support Network. And we act a bit like a sounding board, so the charity can ask us about ideas they have for programs for engaging volunteers and some of the aspects of the research that they do. And we help to shape the volunteering opportunities that many of you have taken part in. And just to give you a little overview of some of the work that we help with, there are, as you know, 3 main themes that the charity gets people involved with volunteering. The first is local groups, so keeping connected, understanding what's going on in research and how you can get involved. The second is the one at the bottom here, actually, about having your say, so that's involvement, being involved in how research is set up, how the trials are run, how patients are included in those. And then the middle one is actually taking part in the research, so being a participant in a trial. And the team helps the staff at the charity to shape all these opportunities that you can be involved in. And the overarching theme that's coming through now is around communication, so there'll be a new steering group set up for that. And as I just mentioned, there are so many ways in which the charity tries to communicate with volunteers, with researchers, with the wider world. And we find in our discussions that we keep coming back to questions of, well, how does it all fit together, and why are we studying this and not that, and how does the charity divvy up its resources between all its different competing demands? So I want to talk you through a bit of work that some of the volunteers have been doing to try and visualize some of the research that's going on and how it fits together. And you've got some of these printouts on your tables, so if you want, you can have a look at them in more detail later on, but I'll just show you on the screen first. So the first is what we've called a research overview. It's a series of bubbles and how they all overlap. So we all know that one aspect of the research is to understand the basics of the condition. What causes it? So work around genes and cell biology and so on. Then you've also got, obviously, the need to find treatments and a cure. So for example, drug repositioning is something that was mentioned already. And then you have all this overlap stuff of things as understanding the basic biology of what's going on, but you can also use that as a route in to try and find a treatment. And of course, the thing that we're all looking for is what's called translational research, actually getting something from the laboratory bench as quickly as possible through to bedside treatments. And this is where a number of the recent initiatives, such as the critical path, virtual biotech that was being mentioned there, where they're so important for accelerating that translation. But, of course, in the meantime, there is the need to continue looking at lifestyle and quality of life. So things such as mood and movement and so on. And again, there are these areas of overlap between treatments and everyday life. And there are complicated things that sit in the middle and seem to be affected. You can look at them from all three different angles. So what we've tried to do with this is just set out some of the big topics that you may hear about and show that they're all kind of interrelated, they're all important, that charity has to try and cover all of its bases. And even at today's conference, we're going to be hearing from people from across this range of research. So first of all, we're going to hear about genetic research from Martin and Miratul. So that's very much in the basic causes area. And then we're going to hear from Heather about drug repurposing, so that's much more on the sort of treatments and cure side. Then we're going to hear from Laura about brain astrocytes. That's again about fundamental research, understanding the causes. Then we're going to hear about dance. So that's very much about lifestyle, quality of life. And then finally, about technology for monitoring and diagnosis. That's very much in this area of what's called personalized medicine, understanding the individuals. So this is just a flavor of what the charity tries to do to cover all the different areas that are also very, very important. And leading on from this, one of our volunteers, Tim King, who's in the audience somewhere, he's put together a really involved and very rigorous map of research. Now, you may not want to go into all this detail, but if you do, he's put down a huge amount of information that you might find interesting, and you might have to take away the printouts and have a look at them, or ask questions at breaks if you can find Tim or one of the volunteers or staff. And without going into any detail at all, I hope it helps to show that there is a lot we need to understand. There's a lot going on, and there are lots of ways of cutting it up. This is just taking 7 big key themes in research at the minute, and a couple of them are more around the causes, a couple at the bottom are around symptoms, and ones in the middle are more about research tools for better studying the condition. But just in a summary, to make it really simple, we need to understand the causes, we need to understand the symptoms and how they come about. We need to develop these research tools like imaging and tracking and so on. And then you have to mix it all up. It's all about trying to understand the condition overall. And you can't just sort of pick a winner and hope it works out. You've got to really keep this broad-based research going on. And then ultimately, when you piece it all together, you get closer towards treatments and a cure. Now, we'd be really interested in getting your feedback about any of these diagrams, the research overview with the bubbles. Is that helpful? The more detailed processes and how they all fit together. If you're interested in commenting on that, that'd be really helpful. Now, I realise that people watching via live stream can't see the detailed pictures, but I'm sure at some point in the future we can circulate them more widely if that's of interest. And if you could think about how they could be used, would this be something that as a lay grant reviewer you'd like to see how the research that you're commenting on, how it fits into a bigger picture? Would that be helpful? Or would you like to perhaps periodically see the development team produce like an update to show, well, which topics have been looked at in the past couple of years and what progress is being made in understanding? And just a final comment. This image here, another member of the development team has a friend who works in graphic design and art, and so they're also looking at ways of how do you make things more accessible to a wider audience. Some people like diagrams, lots of lines and boxes and complicated stuff. Some people engage more visually and artistically. So could the diagrams be made into something based on a picture of the brain or neurons or something? How do you help people to engage in different ways with the science and the research that's going on to make it as accessible as possible? So thank you very much for your time. And I look forward to speaking to some of you later on.
Thank you, Simon. So yes, there are post-it notes on your tables. If you want to make any comments about the maps that Simon's just spoken about, please pop those on and we'll collect them up later. And it'll be really interesting to hear your thoughts on it. And now I'm going to hand over to Martin Taylor, who's also a member of the Research Support Network and in the development team, and Miratul Muqit, who are going to be talking together. Thank you.
Thanks, Anna. I've had a bit of a persistent cough recently, so apologies if it comes across like Theresa May at the Conservative Party conference at times, but I'll give it my best anyway. So my talk today is on a patient's perspective on genetic research. Now, just first of all, just going to get the right slide first of all. A bit of background about me, first of all. I'm 35 years old. I've been diagnosed for 3 years now with young onset Parkinson's. Just done the first thing my wife told me not to do was touch my hair there. Apologies for that. There's another thing there, I said which I've just stopped doing as well. I did a rehearsal last night, which went disastrously, so it goes better. So essentially, I've been diagnosed for 3 years now with young onset Parkinson's disease. I don't normally shake, that's nerves, so apologies for that again. So my journey started 3 years ago. I got married in April of 2014. I was picking up a coin by my bedside and I initially noticed a slight twitch in my right index finger when I picked up a 20p coin. And went to see a neurologist off the back of that. And around 7 or 8 months later, saw another neurologist who, as soon as I walked into the room, said, "You've got Parkinson's." So I was like, what? It can't be right, I'm only 32. So you follow that initial meeting with a conversation right along the lines of what you see in the slide just now. Mr. Taylor, you have Parkinson's, probably idiopathic. Idio what now? It means there's no known cause. Blank stare on my part. My colleague in Dundee is doing some research into genetics. I could refer you, but it's probably not worth it. Right, okay, if you think it's not worth the hassle, I won't bother. Now, there's a presentation in itself as when a patient's ready to participate in research. I don't blame my neurologist, who's a great neurologist, I must say, from dissuading me at that time. But I just wanted to give you a bit of background as to how how that conversation went at the start, with a view to when it's, giving you some feedback on when it's right to participate in research yourself. The idea is very individual. It's very much up to the individual when they feel ready. But the upshot from this is the colleague in Dundee turned out to be Miratul, who I was eventually to encounter 2 years later. So it's just one of those kind of coincidences in life. So, as I mentioned previously, I was diagnosed at 32. I'm now 35. I've since become a dad since my diagnosis to a 16-month-old boy called Cohen, who's doing very well. My symptoms are primarily right-sided rigidity and poor pronunciation. I I've had slow progression so far, and I'm quite responsive to medication. Now, you may wonder why that is of relevance. Well, the genetic form of the disease I potentially have through a Parkin mutation is very much along the lines of these characteristics. It tends to present at young onset, typically in the 30s. It's characterised mainly by rigidity. It has slow progression. It's quite responsive to medication. So I guess I think the slides are out of order here. Apologies for that. I think I'm missing a slide here. I'll go off my notes. So the slide I'm missing basically says the RSN conference 2016. At that conference, one of the presenters was Professor Dario Alessi, who's one of Miratul's colleagues in Dundee. Dario is a world-leading researcher in the field of the LRRK2 gene, and he did a very technical presentation last year on the research that Dundee are doing into that. We got talking after the conference, and he mentioned to a colleague in Dundee, researching onset Parkinson's. That conversation led to a meeting, which led to a test, which led to an interesting result. I guess one of the takeaways from that is this is the research support network. Use this as an opportunity to network. Get talking to researchers, and you might find there's an outcome that can lead to further understanding of the disease, both from a patient's perspective and from the researcher's perspective as well. So you followed that conversation with Dario last year. He mentioned his colleague Miratul in Dundee. So I got in touch with Miratul and went up to see him in January 2017, and we did a blood test. And the blood test came back with a mutation in the Parkin gene in a unique place. So yeah, I got an excited phone call from Miratul in around April time. It sounded almost like it had a eureka moment, and like they discovered this mutation in my gene, and there's going to be further work done on that, and it's not certain it's causing Parkinson's, but there's a fair chance that it's involved somehow, and there'll be a scientific paper published off the back of that either later on this year or potentially early next year. So, genetic forms of Parkinson's disease. There's a couple of slides missing. I must apologise for this. Not all Parkinson's is thought to be idiopathic, is the main takeaway. There are several known genes thought to play a role. Among them are GBA, LRRK2, PINK1 and Parkin. These are genetic risk factors. They're not enough on their own to develop PD. There's an example of twin brothers, for example, with the LRRK2 mutation, both identical twin brothers. One developed the disease, one didn't. So the genes on their own are not necessarily enough to cause the condition. The clinical picture can be driven about where the mutation is within the gene as well. But I guess the main takeaway here is that not all Parkinson's is idiopathic. And there is some suggestion that there's like a known driver for it at the genetic level. So what's the Parkin form of Parkinson's? I must stress again, there's still work to be done to confirm this is the form I have. However, from what I've learned from my interaction with Miratul and my reading around the subject, it's a genetic form of the condition caused by mutation in the Parkin gene. Typically inherited from both parents, although in my case, it differs that it's on a single side, so it's not autosomal recessive. It's typically diagnosed in the early 30s, early to mid-30s, characterised primarily by rigidity, progresses very slowly, and progression to dementia is very rare. It's also very responsive to the existing Parkinson's medication. A unique characteristic of it is sense of smell is very rarely lost as well, which is handy when you're changing nappies, as I have to do on a regular basis. So what's next? A PhD student has been assigned to characterise the gene, genetic mutation, a scientific paper will be published potentially later on this year, if not early next. And it's going to increase understanding into potential genetic cause of the condition with clinical implications further down the line. Now, genetic research is very exciting because it's got potential clinical impact. Now, LARC2 is probably the closest, as I understand it, to being clinically relevant to patients because there's drugs scheduled to go into clinical trial in the next year or two which inhibit the gene. In the case of an LARC2 mutation, which is typically associated with older onset Parkinson's, the gene is overactive. So what they're working on is to have drugs that inhibit the activity and thus potentially slow or stop the progression of the disease. With Parkin, the gene is underactive, so it would need to be reactivated. So that's at probably an earlier stage of clinical relevance, but there is potential for small molecule compounds to be developed which can reactivate the gene, again, potentially slowing or stopping the progression of the illness. So I think a question we'll always ask from a patient community is what relevance does this have to us today? Yeah, there's obviously, in the near future, there's real potential for this to be a game changer in Parkinson's research with clinical impact in a timescale relevant to those of us with the condition today, not just the quote-unquote future generations that we all hear about that are 5, 10, 15 years away. So I guess the main moral of my story here is a positive story of patient participation in research. So like, as I mentioned previously, this is the research support network. So place emphasis on the network part. There's researchers doing presentations today, researchers in the room, get talking to them at the breaks and stuff. You never know what might come from that conversation. And so generally speaking, through patient involvement in research, it's a two-way street. It gives the patient a sense of empowerment through increasing their own knowledge and understanding of their condition, but also feeds into the researchers' understanding of the condition. So there's a lot of benefits to be had from events like this. So make the most of it, and you never know what might happen. So thank you very much for listening to me. I'll hand you over to Miratul, who's going to give a more technical, in-depth explanation of his research into the Parkin gene. Thank you very much.
So thank you very much, Martin, for really introducing this collaboration that Martin is doing with my laboratory. So I'm based at the MRC Protein Phosphorylation Unit, which is one of the world's leading units for understanding how signaling pathways, so these are the highways in cells, go wrong in biology and also in human diseases. And we hope that if we understand better how these highways are controlled, this can start to give us ideas about how to target this for therapies. And when I first met Martin, I met him in one of my clinics at the hospital, and then we invited Martin to come to our laboratories to see how we try and understand more about Parkinson's disease through more molecular and biochemical technologies. So my laboratory has been interested in the Parkin gene, and I have to say that we've benefited tremendously from the work of many genetics labs around the world that have found the Parkin gene. I'm just trying to my slide won't apologies for this, I think. Okay. Yeah, okay, thanks. Sorry, apologies for that. So, over the last 20 years, many genes have been discovered that can cause Parkinson's, particularly in familial cases. And these have been landmark discoveries because they tell us, it gives us some idea of where in a cell the problem lies in causing Parkinson's. And this is a sort of a schematic of a brain cell. And brain cells consist of different components that are required for the health of that cell. And one of the components is this little structure here, which is called the mitochondria. These are the energy-producing batteries of your cell. And several of the genes that have been discovered in patients map to this particular structure. So we know that if you have a fault in any of these genes, then somehow these batteries aren't working as efficiently, and so the cell doesn't get the same amount of energy, it can't protect itself over the long term. And so what our research has been focused on has been how do these genes actually control the survival or the health of this structure, the batteries. And over the last couple of years, my laboratory and others in the field have uncovered a pathway of what's happening in these batteries and how these genes can coordinate that. And so what we and others have discovered is that these batteries can go wrong, and if they go wrong, then that acts as a signal to activate one of these Parkinson's genes called PINK1. And when this PINK1 gene is activated, this becomes a switch for another gene, and this is the Parkin gene that Martin's carrying a fault in. And then once this Parkin gene is also activated, then it makes a particular type of molecule in the cell which is protective. So this is a pathway which is ultimately protecting all of us from developing brain cell loss that could be leading to Parkinson's. And what we think is happening in patients is that if you have a fault, so if you inherit a mutation in the Parkinson's gene, then this protective pathway is disrupted. You know, it's lost, and these brain cells can no longer cope with stresses. So what is the Parkin gene? So the Parkin gene was first discovered in 1998 by Japanese researchers. It's a very common cause of early onset Parkinson's. So clinically, it means that if a patient comes to the clinic under the age of 21, there's a 90% chance that they have a mutation in the Parkin gene. And even in patients under the age of 40, it's a very important cause. So it's probably estimated about 40% of patients who develop Parkinson under the age of 40 have Parkin. And it's normally, you know, the inheritance, and Martin touched on this, it's what is called, it's inherited in an autosomal recessive fashion. So what that means is that usually the parents each carry, you know, one healthy copy of the Parkin gene and one faulty copy of the Parkin gene. And if you inherit, you know, if you inherit 2 copies of the faulty gene, then you could develop Parkinson's. And usually having 1 copy is not sufficient to cause Parkinson's. And that is just generally a general principle in genetics. But in Martin's case, Martin carries only 1 change in the Parkin gene. And ordinarily, you know, this would perhaps not be significant. But in the clinic, there are many patients like that Martin who carry these single changes, many patients who are young. So this raises the question that, you know, maybe in Martin's case and in other patients with similar changes, maybe having 1 fault in the Parkin gene could lead to Parkinson's. And that's what we're investigating. That's what the collaboration's about. Now, what is the impact of Martin's change from what we know about the gene? And so a lot of work has been done on this pathway, and this is an image of the atomic structure of Parkin. This has been solved by many groups around the world, including by one of my colleagues in Dundee, Helen Walden. And you can see here in this diagram that the Parkin gene is very complicated. It has lots of loops, and it has lots of sort of spirally type of elements. This is, you know, at the atomic level, how this enzyme is being controlled. And where Martin's changes is he's right over here. And I've just taken this region here and I've blown it up. So Martin has a change in a residue, an amino acid, called proline 437. And you can see that this residue is important because it's keeping this part of the gene in a particular conformation. You can see there's a sort of a kink here, and that residue is keeping it in that conformation. And we would predict that Martin's change that he has would affect that. So just by looking at the atomic structure, we would imagine that the change that Martin has would lead to some effect on the gene, and that is what we have gone on to investigate. So how are we doing that? So what we're doing at the moment are experiments using a variety of systems. We're expressing this Parkin gene in different ways, in bacteria, in human cells, and then we're trying to then see what its activity state is. And what I'm going to show you next is the first piece of data that we have from this project. And so I'm going to walk you through this in 3 steps. So what we want to know is if we activate Parkin using this pink 1 switch, we can monitor it. We can monitor in the test tube, and that is what I'm showing here. So this is what Parkin is like normally. It's normally switched off, and you can see here you can't see any signal. You can't see it making any of these chains. If we add pink, then you get this Parkin being switched on, and you can see here very clearly that you get these very, very prominent ladders of molecules forming. So that's what happens if we have healthy copies of Parkin. What we can do is we can engineer the Parkin in the laboratory so that it's not what I'll do is I'll just switch this first. We can engineer it so that it's not working. So this is a patient whose Parkin isn't working at all, and you can see here that when you add the pink, you don't see any laddering whatsoever. So this is somebody who clearly has a clear fault. So what happens now with Martin's change? And so what we see here actually is that very surprisingly, because we didn't think this from the atomic structure, very surprisingly, Martin's change doesn't seem to affect the ability of Parkin to make these molecules. So this is actually quite an important result. It suggests actually that what we know so far about the Parkin enzyme in terms of what it's doing may not be complete. It may be because of the fact that Martin's change is active in our tests. It may be that there's actually more to understand about what Parkin does, and this is going to stimulate further studies. And certainly, I think at the moment we can't conclude at the moment whether Martin's single genetic change is really causing his Parkinson's, but this really, I think, has stimulated a new area in the lab to really understand more. So what's next? So what we're doing is we want to use Martin's example to really address this issue of other people that have other changes, and we want to set up a platform where we're looking at lots of other changes in the Parkin gene and seeing how they affect it in the laboratory. And then what we're also doing in Dundee is that some of you might have had genetic testing on the NHS. And normally, at the moment, anywhere in the UK, you only get 2 genes tested. You get the Parkin gene and you get the LRRK2 gene. But one of the things that we want to do in Dundee, which is underway, is we think every patient should have all the genes tested. So there are 18 genes that cause Parkinson's. And Martin's sample is being analyzed at the moment for all 18 genes. And Martin will get the result of those 18 genes. And we will be able to understand whether maybe we find another change in another gene in Martin, which is much more decisive. But if we don't see any changes in Martin in these other 18 genes, then it may, again, suggest that this Parkin change that he has is significant. So I think what this overall has done is it's really stimulated a lot of developments in Dundee, both at the kind of experimental level, but also at the clinical genetic level, because it's made us reappraise how we test patients and the fact that we should be more comprehensive as well in gathering all the information and relaying that back. And I think overall what this will mean is that patients coming to the clinic will get, they'll get a full picture of their genetic landscape. And this will be very important because in many fields of medicine, we're heading towards personalized medicine. In cancer, particularly, people are being diagnosed and treated according to their genetic characteristics. And this is, I think, the goal of Parkinson's and other related neurological disorders. And I think these sort of initiatives will really help towards that. So I want to finish off by mentioning those in my laboratory who are doing the work. So Christine Balk is a PhD student who's working on Martin's change, and she was assisted this summer by Clara, who was a summer student visiting from Barcelona. And then Atul is a structural biologist in my lab who has worked on the Parkin. Thank you very much.
Thank you very much, Martin and Miratul. So I'm now going to welcome Heather Mortiboys from the Sheffield Institute of Translational Neuroscience. There's a mouthful for you. Thank you, Heather. And this is our last session before the break.
So, thank you guys, and hopefully I won't keep you too long before you can go and have a break and a cup of tea or coffee. So I'm going to talk to you today about a project which has been ongoing in Sheffield since 2013 in my group, and that's along the drug repurposing strategy that both David and Simon introduced earlier on. So just very briefly, drug discovery in Parkinson's, and we've already heard that there's kind of two basic routes that this can be done along, developing a new drug with completely new chemistry, and this can be time-consuming and expensive, and in terms of the pharmaceutical industry, can take anywhere between 10 to 20 years and cost upwards of $800,000, if not $1 billion. And as David alluded to earlier, this has meant that certainly some pharmaceutical companies have in fact reduced their input and their work they're putting into drug discovery for Parkinson's and other related conditions. But there are already more than 14,000 drugs which are known to be safe in people and they're known to be effective for particular conditions and they're prescribed clinically. And so we can take the drug repurposing approach, which is by investigating these 14,000 drugs to see whether they in fact could be effective in Parkinson's and bring a new treatment to people with Parkinson's more quickly. And that's the approach that this project has taken. It's focusing on a drug repurposing approach. So the part of the cell that the project really focuses on is the mitochondria. And usefully, both David and Miratul have introduced the mitochondria to you. So they're the batteries of the cell, and they're contained in all of our cells of the body. Not just the neurons, the brain cells, but in our skin cells, in our muscle cells. They're present in multiple copies in all of our cells. And I've put them here in a little cartoon picture of a neuron as these kind of ovals. And in some cells they do have that shape, but actually they're really dynamic, and they can move around the cell to where energy is required. And they can change their shape and come together into fusion and then divide into fission, dependent on the energy requirements of the cell. And a bit like us, they can use different types of energy, different types of food to make their energy. So we know that if we have a particularly sugary drink, we'll have a different energy level than if we were to have a bag of chips. And that's because our body can use those different foods in a different way to generate energy. And the mitochondria in our cells are exactly the same. And ourselves in Sheffield and many other groups around the world have spent a long time looking at mitochondria and Parkinson's in Parkinson's patients. So in the brains of Parkinson's patients, and as David said, he was in the group that first really found this, that mitochondria didn't work properly in the brains of Parkinson's patients. But subsequently then, we and other groups found that the mitochondria don't work properly in the peripheral tissues of patients with Parkinson's, and that includes muscle and blood, and also skin cells. And then, as Miratul alluded to, if we are to make a genetic model of Parkinson's, either with the Parkin gene change or with the LRRK2 gene change or any of the others, and this might be a fly model, a zebrafish model, or a mouse model, pretty much across the board, these animal models also show mitochondrial abnormalities. So whether we look in patient cells or in these ways that we can model Parkinson's, the mitochondria are not working properly. And so what we did was we used this information and this knowledge that we had to design a drug repurposing screen. So the first question for us was which drugs were we going to screen? And as I said, we decided to take the drug repurposing approach to take these 14,000 drugs, which are already used for other conditions. However, in Parkinson's, ideally we want some of the drug to get into the brain, to the cells which are lost, and this means that they have to cross this blood-brain barrier. So what is the blood-brain barrier? So here there would be a blood vessel in the brain, and it's surrounded by these barrier cells. And then after the barrier cells, then you have the different types of brain cells. And so the drugs which you would take in a tablet would get into your bloodstream, and then they need to get past these barrier cells to actually get into the brain cells. So on these barrier cells, they have active transporters, and some molecules like this one will be taken up by the active transporter and be able to get through the barrier cell and then have access to the brain cells. Some of them, like this white one, are small enough to squeeze through those gaps in the barrier cells and again then be able to gain access to the brain cells. But some, like this one here, is too large to fit through the gaps between the barriers and isn't taken up by those active transport mechanisms to get through the barrier cells. So this drug would not get in very large quantities actually to the brain cells. And so what we did was we used a technique on the computer to rank these 14,000 drugs for their ability to actually make it to the brain cells where we need to. And then we only took the top percentage of drugs of which we had good evidence to say they would get into these brain cells to testing in the laboratory. And so then what did we actually do in the laboratory? So we utilised the skin cells from patients with Parkinson's, which, as I said, we'd spent quite a long time previously really showing that these skin cells, their mitochondria were not working properly. And what we do is we've taken skin cells from 3 groups of patients. From patients that have the Parkin mutation causing Parkinson's that Miratul and Martin introduced, from patients that have the LRRK2 mutation causing their Parkinson's, and then from the largest group of patients that have sporadic or idiopathic Parkinson's where there's no identifiable cause. And in all 3 groups of these patients, the skin cells that we're testing, we've already identified that the mitochondria are not working properly in them. And so this is Hannah, who works in the group, and what she's doing there is she's plating skin cells into these plates. So I'm afraid I've only brought one of them with me, but as I've got the tea break after me, then you're more than welcome to come and have a look at them. So these are plates that have 384 wells in them, and she's putting skin cells from patients with Parkinson's into each individual well of those plates, and then we can put them in the incubator in the laboratory. The cells will stick to the bottom of the plate, and then we can use them for the screen. So after that, the drugs are then added, and we do this using a robotic system that enables us to dispense down to extremely tiny amounts of drug, very, very, very quickly and with great precision. And it actually dispenses a millionth of a teaspoon at any one time into each well of those plates that have 384 wells. It's much faster and it's much more accurate than I am doing this. And then what we do is we take those skin cells that have had the drugs on them and we stain them. And we stain them for 3 things. We stain them for their nuclei, which is the DNA organizing center of the cell, for their mitochondria, and we also stain them for their lysosomes. So the lysosomes are the waste disposal route of the cell, And there's also a lot of evidence to suggest that that pathway, that waste disposal pathway, isn't working properly in Parkinson's. So once we've done this staining, we then use a high throughput microscope, which takes 20 pictures of the cells per well of the 384 well plate. And then we can take those images and we can look at various different parameters, the size, the number of the mitochondria, how bright the staining is, which tells us how they're working, and the same for the lysosomes. And this is some of the images here. So you can see the different types of staining. The images at the top, the mitochondria, are in green with the nucleus in red. And the whole screen actually generated more than 2.5 million images for us to analyse. Fortunately, we managed to automate that process and didn't have to do it manually. And so what did we find? So we did that process across the cells from the 3 different patient groups, and we identified several groups of hit or lead compounds. So we found 21 compounds that preferentially had a beneficial effect on the mitochondria only, and they didn't have an effect on the lysosome pathway. And we found 4 compounds which only had an effect on the lysosome pathway, but no effect on the mitochondrial pathway. And then we found 8 compounds which actually had a beneficial effect on both pathways, the mitochondria and the lysosomes. And really importantly, all of these compounds here were active across all 3 patient groups. So they were active in the cells from patients that have the Parkin mutation, from cells from patients that have the LRRK2 mutation, and from the cells from the patients with sporadic or idiopathic Parkinson's. In addition, it's also important to note that actually we found a group of compounds that only were active in the cells from patients with LRRK2 mutations. They didn't work at all in the Parkin or the sporadic group of cells, but they were pretty much the most active compounds in the LRRK2. So we're actually taking some of these compounds as well as this group of compounds forward into our next stage of what we did next. So this was then the secondary screen, and what we wanted to do is, are these compounds really having a positive effect? And we can test this in some different ways. We first of all checked that they were active over a range of concentrations, and then we looked at alternative laboratory methods to check that they were having a beneficial effect on the mitochondria and the lysosomes. And then we really then went on to test how long it took them to have this positive effect. So our original screen had been at 24 hours of drug treatment, but we wanted to see, do some compounds already have that beneficial effect after an hour, or do some actually really need that 24 hours to have that beneficial effect? And this is just some of the results from that. So here on the left-hand side, we've got some cells from patients with sporadic or idiopathic Parkinson's, and this is looking at a mitochondrial measure. So down here at the lowest drug concentration, then the cells are not producing very much energy. And as we increase the drug concentration going across here to the right, we can see that we can increase the amount of energy that the cell is producing. And here the compound in green increases the amount of energy the cell is producing up to this level, which is about 95% of where a control cell would be, whereas this compound in blue only increases it up to about 85% of where a control cell would be, which is still a particularly active compound. And then here on the right-hand side, this is again some cells from a patient with sporadic or idiopathic Parkinson's. And what we've done is we've added these different compounds for different amounts of time. So time now is along here. And so we can see that the compound in red already, after 2 hours, has shown a massive increase in the amount of energy that the cells can produce, and then goes on to have another small increase when it's dosed for longer, but already there's a very clear effect after 2 hours. Whereas this compound in the light blue, it really takes the full 24 hours of incubation with the cells to have that positive effect. And that's really going to tell us something about how those different compounds are having that beneficial effect on the mitochondria in this case. We're also doing the same for the lysosome pathway, but I won't show you that data today. So all of that work was done in the patient's skin cells, but what about the brain cells that are actually lost in Parkinson's? So what we did together with Laura Ferraiuolo, who's giving a talk after the tea break to you, is we turn the patient skin cells into brain cells. So Laura really pioneered this technique. So what she does is she takes the skin cells from the patients and she adds some reprogramming factors to them. And this will fool the cells into thinking that they are not skin cells anymore, but then in fact they can become other cells. And so if we add a certain amount of factors and grow them in a particular media, then we can turn them into a particular type of stem cell. So a stem cell is a type of cell that can become lots of different types of cells. And what we do is we turn them into a type of stem cell that can become different types of brain cells. They can become any type of neuron, and they can also become the supporting cells in the brain, astrocytes that Laura's going to talk to you later about, or oligodendrocytes. And then what we can do is we can have a different set of factors which will really make them into the type of brain cells which are lost predominantly in Parkinson's, the dopaminergic brain cells. And then when we've got these, we can add the drugs to see whether they have a beneficial effect on their mitochondria and their lysosomes. So we've turned the patient skin cells from our 3 groups of patients into brain cells. And then we've tested the drugs to see whether they actually have a beneficial effect or not. And the answer is yes, they do. So this image here shows you some of the brain cells that we can grow in the laboratory. These ones are from a patient that has a Parkin mutation. And here there's lots and lots of cells, and you can see them forming connections and networks with each other. And here again, we've stained for the mitochondria and for the lysosomes, and we can get similar types of readings and measurements from these brain cells as we did from the skin cells, and really find out that yes, the drugs that we identified in the skin cells are having the same beneficial effects in the brain cells. So what's next then for that study? So to summarize, firstly, we've found drugs that improve the energy and the waste disposal route of the cell. So they actually target 2 different pathways. And all of these drugs are repurposing candidates, so they're already known to be safe for use in man. So the next steps, they could be a variety of different things, and a lot of that will depend on where we want to take this further. We could be required to test some of these compounds in various animal models of Parkinson's, although, of course, we do know that they're safe to use in man, so if we could show enough of an effect in the cells from patients, we may not have to do this. Probably very importantly, the way that we designed the screen, we designed it so that it was looking at pathways. It was looking at the mitochondria and it was looking at the lysosomes. So we know that the compounds have a beneficial effect on those pathways, but we don't know exactly where in the cell they're having that beneficial effect. We don't know what part of the mitochondria, or in fact, if anything, they're not going to the mitochondria, but they're having an effect on another part of the cell, which then causes an increase in the mitochondrial function. So that's really important for us to determine, and that's where a lot of our efforts are going at the moment. In addition, thinking further ahead, and if we were thinking about a clinical trial for any of these compounds, then really, ideally, what we'd like is to be able to determine if we can track mitochondrial activity and lysosomal activity in real living patients as they were taking the drug. And that would be fantastic to enable us to design a clinical trial. And as David alluded to earlier, this is something that other groups around the world are progressing forward. We would then obviously have to determine the best drug to take into clinical trial, because we still have around 10 compounds which are being active in our brain cells, but we'd need to decide and focus in on one compound that was going to be the best one to take into a clinical trial. And we need to be in discussions very, very early on with different funders of clinical trials to see what they would require us to do to really show that, that these compounds are worthy of being taken into a clinical trial. So finally, I should say thank you to everyone who was involved in this project. So you saw Hannah doing the plating of the cells, and she really did the first stage, the first screening of the project, and then Aurelie came into the group and has taken it over since then with the help of Camilla and Martin was really the person who did the computer-based screening right at the beginning of the project. And just finally, oh no, James, it's not there. So there's no slide, but finally, we at Citran, we have an image competition that's going to go live on Twitter on Monday morning. And so this went out to all the PhD students and the postdocs in the laboratory to use some of our high throughput microscopes to take the best, prettiest picture of their science. These were shortlisted, and there's 4 now in the shortlist. Actually, 3 of them are from people in my group, and the 4th one is from someone in Laura's group. So we both have a vested interest. And the winner will get £500 to go to a research conference. And so if any of you are on Twitter, then if you look for the Citran, or I'll be retweeting it as well, and then it's voting for the best image, and then we'll see who the winner is a week later. Thank you very much.
Yeah, thank you for the introduction, and it really is a pleasure to be here and share with you some of the data and the future direction of a Parkinson's UK funded project looking at brain astrocytes, so a different cell type in the brain that is kind of like helping the neurons. And this is a project totally co-led with Heather Mortiboys, who presented just before me. So first of all, I'll give you a bit of background about me. I think you got me from hello, I'm Italian, and I studied my undergraduate degree in Italy, Milan, and my course was in biotechnology, so already I was studying a bit how we can kind of like manipulate and modify cells. And during my internship, I was in a laboratory studying neurodegeneration, and in particular motor neuron disease. So then I moved to Sheffield for my PhD, and that is when really I got deep inside motor neuron disease and how we can use cells to model neurodegeneration in vitro, so in a dish. And then I went to Columbus in the States. It's in Ohio, just under the Great Lakes, and I was there with a EU-funded fellowship. Basically, the aim is to go abroad, learn something new, and bring it back to a European lab, which for me was again Sheffield. And so the technique I learned is actually really the basis of our research and what we're doing at the moment. And then I came back, as I said, I went back to Sheffield, and here I'm setting up pretty much my laboratory, my research group in collaboration with several partners within the university. So today I'll be talking a bit more about kind of like neurodegenerative diseases in terms of how we think of them in terms of neuronal vulnerability. So doctors, medics have always looked at neurodegeneration as something that affects the neurons because patients show up with symptoms that really shout basically neuronal death. So neurons are the cells in your brain that regulate your movements, regulate your thoughts, your memory, your coordination, and therefore when a patient shows up in clinic and has lost some of the motility or memory, cognition, and so on, obviously it has to be because neurons are affected. So neurons are the star of the show, really, as you can see here. However, if we think of the brain is a collection of, yes, 86 billion neurons, but actually there are even more non-neuronal cells. And these non-neuronal cells altogether, we call them glia, which is a very unfair term because it basically means glue. So in the past, researchers just thought glia was there just to keep together the neurons, pretty much. But it's not true. So as I said, like when we think of the brain, we always think of it in these kind of like beautiful terms. These are the neurons, these are the cell bodies, and these are the long projections they shoot out to kind of like muscles, other neurons, and how they make connections. And Heather actually showed some beautiful pictures of how even in vitro, even in a dish, they make connections, and that's the basis of how they work. But if we zoom into this picture, this is actually what we see. And you can see that if the neurons are in blue, Actually, there's a lot of other cells in red and in green that are not neurons, and that is glia. So specifically, who are they? I'm going to introduce them. So the blue are the neurons. And then in red, we have the astrocytes. And the astrocytes, they really are the star of the show because astrocyte actually means a star. The microglia, the green cells that are also called, as I said, are also glial cells. So these are the neighbors of the neurons. But the question at this point, I told you they're very important, but what do? They do housekeeping. That is really what they do. So if you think of your brain as Buckingham Palace, and if you think of the neurons as the queen, very high maintenance, then the glia, are all these people who are basically like cleaning the house, preparing the food, providing the food, serving it at the right time, in the right amount, so that we don't end up having a fat queen. And so they really kind of like coordinate all of this. So you can understand now at this point why they are so important. Why, if the glia are not working well, then the neurons are not working well, and they don't live in a good environment. So I'm going to show you some more kind of like practical images of what these cells look like when we manage to isolate them and put them in a dish. And so you can see this is, so the green cells are the neurons, and the red cells are the glia. So this is a movie, and this is kind of like a, let's say, healthy scenario, so where the glia are working fine. Okay, so you can see that they are moving. Here they're moving in slow motion, but actually they move quite fast. And you can see they go around, and they go around the neurons, and they check that the neurons are fine, that their projections are long and healthy, that the cells are connected, and so on. You don't see anything kind of like aggressive or dramatic. They're really just housekeeping, checking that everybody's happy. And if there are small debris of neurons, so they basically kind of like just absorb them so that everything is clean. Now, if we go ahead, I'll show you a scenario where we have glia that is not healthy. And in this specific scenario, the glia is from a person with a motor neuron disease. So this is kind of like a different scenario. Again, neurons in green, glia in red. And if I may give you a spoiler, look in the middle of this video. But you can see immediately that kind of like the glia are moving a bit more frantically. Pick up. They're moving a bit more frantically. If you look up there, you can see that they are pulling the projection of the neuron. And also the neurons are a lot more motile because they're not happy in this environment. But again, if you keep kind of like focusing in the middle, you can see that now the projection there has been snapped pretty much. Becomes first long, it gets snapped, the neuron disappears, and then you'll see that now down here, the other neuron will disappear. So it's basically like in Buckingham Palace, there's a lot of undercover assassins, pretty much. And again, you can imagine that is not a good case scenario. So now you understand why we are studying glia and why we think it's important. So how do we study kind of like human, both neurons and glia? Again, Heather gave you a spoiler. I'm just going to go a bit more into details. And that takes me to this scientist, Shinya Yamanaka, who won the Nobel Prize in 2012 because he really revolutionized the way we can model diseases in the laboratory, and especially neurodegenerative diseases, because having access to the brain when people are still alive is close to impossible without creating an extensive amount of damage. So that is a very well-deserved Nobel Prize. And the technique, as Heather already told you a bit about, basically starts from skin biopsies. So doctors just collect a little bit, a punch of your skin, and then we can grow skin cells, fibroblasts out of those. And then with the technique developed by Yamanaka, you add some reprogramming factors, which are, you know, again, as Heather said, a bit Trojan horses that convince these cells within 60 days that they are not skin cells anymore, but they are stem cells. And the stem cells are basically those cells that are in embryos that give rise to the 220 different specialized cell types in the body. So you can understand how revolutionizing kind of like this technique and this discovery was. So we have taken this technique. This is what I did when I was in the States. We took this technique, and I don't think we are going to get the Nobel Prize for it. However, we managed to engineer it in a way that, again, we take skin biopsies, and then we grow skin cells. And not within 60 days, but within 30 days, we have a cell type that cannot give rise to 220 different cell types. It can only give rise to the cells that are in the brain, so neurons and astrocytes, so the glia. But this is all we need, really, at this point. We shortened the protocol and made it in a way that was useful to us because we wanted to model what happens to neurons and astrocytes. We really didn't want to necessarily make liver cells or other cell types in the body. So, and that is the reason why this protocol is shorter. So then the question we were able to ask at that point with Heather was, are astrocytes then dysfunctional in Parkinson's? When we take cells from people with Parkinson's, do we see the same scenario that I showed you earlier in the video, like what we see with motor neuron disease? And so we focused on Parkin, and now you become experts in both Parkin and mitochondria. So Parkin, a bit more in general terms, is involved in the disposal of damaged mitochondria, which are the cell power of the cell's powerhouse. And as I said, you are now experts, so I don't need to go in details of that. And we know that dysfunctional Parkin actually causes abnormal function of mitochondria, or abnormal handling. And in fact, people carrying mutations in Parkin develop Parkinson's. Again, we heard about the genetics of it. So what we did then, we used our donor-derived samples. We took skin biopsies, and here they are, the skin cells. We reprogrammed them, and we made astrocytes. And these are the beautiful cells that got in the competition you need to vote for. Aren't they beautiful? So we took these astrocytes from people with Parkinson's and people without Parkinson's, and we ran some assays. And what we asked is, how many mitochondria do these astrocytes have? So, you know, are they set to produce the right amount of energy that they need? And what we found is that actually, if you look in this graph, so this is the number of mitochondria that you have in people without Parkinson's, and this is a group of people. And let's say they have about like 100 per cell. And then if we look at our volunteers with Parkinson's, and these are two representative people, we are looking at more individuals, they have more. And this star actually means that mathematics, so statistics, gave us the stamp that it's true. We're not seeing this by chance. This is actually significant. It's actually true that these cells have more mitochondria. So then intuitively you think, okay, if they have more, that must be good. So we asked, well, actually, is it? So how hard do they work? Do we have more and they work equally well, so we have more energy, or they work less? And actually the answer is that they do work less. So if you look at this graph, this is a measure of how hard the mitochondria work. Again, mitochondria from people without Parkinson's, let's say, are set around 100%, and then people with Parkinson are actually lower. They work about kind of like 30% less. So again, if we kind of like go back to our simile with the housekeeping and so on, it's like having basically 30% of people in Buckingham Palace are not working, and that obviously cannot be any good. So where are we taking these next? So in summary, what we think is happening is that in Parkinson's astrocytes, we do have more mitochondria, but they are less fit. And therefore, that means that we have less functional astrocytes. On the other hand, we know that because these astrocytes are from people carrying a Parkin mutation, probably this is due to the defect in disposal. So what we have is actually accumulation of damaged dysfunctional mitochondria. And again, this leads to kind of like dysfunctional astrocytes. So again, we said how important glia is, but then what is happening to the neurons that are surrounded by this glia? And so what we are doing next is actually to assess the effect of Parkinson's astrocytes on neurons. So what happens to these healthy neurons if we put them in culture with astrocytes from people with Parkinson or astrocytes from people who don't have Parkinson's? And this is an open question. At the moment, I don't have data. Maybe next time I'll show you another video. But this is basically what we are doing at the moment. And then the next step again for us is actually if we take these damaged, unfit mitochondria within these unhealthy astrocytes and we treat them with the drugs that Heather has identified in her screening. So we don't have to go back and rescreen everything, but we take just the best hits. Do we manage to make stronger, healthier mitochondria and therefore more functional astrocytes that are going to support the neurons better? And so I hope by now you have an idea why this is important, but it is important because we really think it's going to kind of like this synergy between this technology and Heather's drug screening is going to facilitate really drug discovery and the next step. So what we think is that this approach is quite powerful because we're going to assess the function of the compounds Heather has identified not only in neurons, but in all the brain cells, in astrocytes as well. And we're going to have a more complete picture of what is happening. Because as I showed you before, even if you have a healthy neuron, but then you have a league of assassins surrounding them, then this is still not going to give us a good result. Obviously, Heather talked a bit about the kind of like preclinical test, and that is the long part of kind of like drug discovery. You know, you need to test that these drugs actually also even when they are repurposed, when they are in high quantity in the brain, do they activate other pathways that maybe shouldn't be activated and so on. So we still need to run, you know, some safety studies. And that can go, you know, on for various amounts of times, and then finally, you know, we'll be able to go into clinical trial. So I just wanted to thank you guys for the attention and for welcoming me into this association, which is really nice. You know, there's no point in me thanking Heather because we are co-leading this, so actually I'm kind of like talking a bit on her behalf as well. And, but we definitely need to thank Camilla Boskian, who's a very talented research technician funded by Parkinson's UK. She has made incredible progress, and actually all the data I showed you and much more has been produced by her. And then Hannah, Aurelie, and Monica helped with the tissue culture and the reprogramming of the cells. Thank you.
Thank you very much, Laura. So we're going to move into the panel session now. If you have any questions based on Laura's presentation, please write them down now and raise your hand, and a staff member will run over and grab that for you. And I also want to take this opportunity to say hello to everyone in the live stream. We've had quite a few people. They're from all over the UK, and we've even got someone in Canada. So hello, and if you have any questions, type them in. Thank you. Right, could we have our speakers up on the stage now? Thank you, David. We don't want to blind our speakers. Excellent. All right, so the first question I have here is for David. from Andrew Leach in the room. How do we get drug companies interested in repositioning, as normally the drug involved is off patent and there is less profit available?
That's a very good question. At the moment, there is no real roadmap to actually get drugs repositioned. Now, if the original use of the drug is totally different for its potential use in Parkinson's, you can do what's a change of use patent. Say if it was used to treat asthma, and then now it's been shown in clinical trials to actually being beneficial for treating Parkinson's, you can do what's a change of use patent. So then the drug company would be interested in that because it's got a bit of extended life to it. But if something has already been patented for that use in Parkinson's, say if they did a blanket coverage, there's very little roadmap there, but we're working with the government agencies to try and get it through, to get it registered into the BNF, which is the doctor's Bible about the list of drugs that can be prescribed and things. But at the moment, there is no real roadmap, and we are working with the government agencies to try and formulate this in advance of any of the drugs being repositioned.
Thank you, David. Next question is for Heather. This has come from Laurel Miller, also in the room. Of the 14,000 drugs tested, of the ones already in clinical trials, such as simvastatin, where do they stand in this analysis?
So simvastatin and exenatide, also in clinical trials, they didn't make it through to stage 2 of the process, and that may be because the screen that I outlined is really looking for something that will target mitochondria and lysosomes as a mechanism and stop the cells from dying. So really looking at it in terms of a mechanism rather than a symptomatic or simply being beneficial for the cells. So that may be why they didn't make it through.
This one is for Miratul and Martin, from Janet Hughes. She says, we were never offered genetic testing, just told that the Parkinson's was idiopathic. How can we gain access to genetic testing in order to establish the type of Parkinson's we're dealing with? Or is this only offered in the case of early onset Parkinson's?
So most centres actually in the UK will have access to a regional genetics laboratory where one could get your Parkinson's genes tested. I think the decision whether to test or not depends on your primary clinician in the sense that so if you have a family history or if you're very young onset, then your primary clinician may think that is, for his point of view, very likely to be more worth doing a genetic test. If you are, when you say idiopathic, if you're somebody who's much older, there's no clear family history, the likelihood then of your primary clinician wanting to do that is much, much less. I think as more genetic knowledge is gained, and not just in the sort of the familial form, but also in the general kind of sporadic form, there's now clear what variants can increase your risk. I think it will take maybe 5-10 years when the genetic understanding is much, much more improved. It may be then that you could, even for a late onset idiopathic, derive some information from that route. But I think at the moment it would depend on your circumstance of family history and age of onset.
Well, worth mentioning as well, you do have the option of 23andMe, which is like a private company that does genetic screening. It will give you like information on your risk variants. It's owned by Sergey Brin, or his ex-wife actually owns it, who's the founder of Google. He carries a LARC2 mutation, and his mother has developed Parkinson's. So he's kind of got a vested interest in the genetics of Parkinson's. So I think it's around about £100. Periodically it's free for Parkinson's patients. I don't think it is at the moment. But that is an option as well, if you want to get a private screening of your genetics. But I would say that anyone thinking about doing genetic testing, don't take it lightly. It's telling you about your very fabric of your being. So you might get some nasty information there that you don't necessarily want to know. So just give it full consideration if you're going to go ahead with that route. yeah, that is a private option if you want to actually pay for a genetic test. You can go through 23andMe.
Thank you. This one's from Andy Hobbs to Simon, more of a request. Can you post the soft copy of the printed material, the map that's on all the tables, for download from the website? And someone else has also said it's really useful. Is it available on the website, or will it be?
So that's something we were hoping to get feedback on today. We're pleased that people find it helpful. I think the team will probably discuss it with staff and just make sure that it's correct in its details, and then yes, be able to make it available in some form. Yeah, we're glad that it's helpful.
We will be sending it out via email to all of you who are here today. And yes, as Simon said, we'll do some checking and see where we go next with it. Yeah. This is one for Laura. Do you see changes in astrocytes in post-mortem brain tissue from people who had Parkinson's?
Yes, so there are kind of like publications that say that the astrocytes, when you look in post-mortem tissues, are activated. So there is some sort of activation. Inflammation, as we call it, is a very general term and general phenomenon that is common to several neurodegenerative disorders. So yes, we see that they are activated. We see that they are not just like in a brain or in a tissue from someone who hasn't had Parkinson's.
Thank you. This one's another one for David, from Peter Miller. Many of the Parkinson's researchers are from EU countries. What are the implications of any type of Brexit, hard or soft, on the availability of researchers?
Yes, another good question. This has been flagged up. The government has written a life sciences industry strategy, and this strategy document does flag up the need to still have a free flow of international scientists into the UK to maintain our compatibility in the market, really. The life sciences really does contribute quite a lot to our economy. Many of our pensions rely on shares in these companies as well, but these scientists are important for keeping research going. And we're going to Downing Street, I think it's the week after, and we're going to be lobbying hard to the MPs there to make sure that the life sciences strategy is actually carried through and it protects the free flow of scientists into the UK.
Thank you, David. We've got a question from the live stream, so that's excellent, from a researcher actually, Simon Stott, based in Cambridge. Heather, can you provide an update on UDCA? Has there been any follow-up work on this?
Yes, so UDCA, so we undertook a drug screen before the one that I talked to you today about, which was focused on finding compounds that were beneficial for mitochondria only, and we started off just in cells from patients with Parkin mutations, and out of that screen came UDCA, which is ursodeoxycholic acid, and that's already in clinical use for primary biliary cirrhosis, which is a liver condition. And so that screen finished, and the paper came out in 2013 now. So since then, we have been testing in other types of Parkinson's. So we've tested it in LRRK2 forms of Parkinson's, both in the skin cells of patients and also in fly models of LRRK2, and UDCA was protective in both of those LRRK2 models. We've also tested it on the mitochondria from some of the sporadic patients' cells that I was just talking about. And UDCA does have a partial rescue effect in those sporadic cells as well. And alongside now trying to get UDCA into a clinical trial for Parkinson's patients, we're also trying to figure out exactly which part of the cell UDCA is having its beneficial effect on to increase mitochondrial function. Because at the moment, that's kind of a black box question. We know it's going into the cell and we know it's improving mitochondria, but we don't know how. And so in parallel to trying to get funding for a clinical trial for UDCA, we're also trying to figure out exactly how it's working in the cell.
Thank you. Here's another one from the live stream. Nicola in East Yorkshire. I know you, Nicola. Hello. Nicola says for Miratul, some of us have genetic tests from 23andMe done via the Michael J. Fox Foundation. Are these useful for your group?
So I think if you've had your 23andMe data analyzed and you have got in the analysis mutations that or confirmation that there is a gene that causes your Parkinson's, then I think this would be of interest to our group. And one of the things that myself and also my colleague Dario Lessi are interested in now is that, so at the moment we have these 18 genes and these all seem to cause Parkinson's, and at the moment they all seem to lie in sort of separate functional pathways. But it's very likely that they exist in networks. It's very likely that there may be crosstalk or communication between one gene and another gene. And we now have very good tools to study the pink Parkin pathway. Dario Lessi's lab have very good tools to study the LRRK2 pathway. So we are actually very interested now in obtaining as many patient-derived cells or samples of patients with different sorts of mutations, because then we can ask them, because you might not have a change in the pink Parkin or the LRRK2, you might have a change in, for example, maybe DJ1 or synuclein, and the question is, do you still have problems with LRRK2 or pink? So yeah, I think if the data has been analyzed and there's a clear-cut mutation, then that could be of interest to us, for future studies.
Thank you. This is another one for Heather from Jane Oliver in the room. As I understand it, one of the main drugs used in Parkinson's, levodopa, doesn't readily cross the blood-brain barrier, but is enabled to do so by combining it with another drug, such as carbidopa. Are you missing out some potential repurposing drug candidates by only considering those drugs that will cross the barrier?
Yeah, so that's that's a good point. That there's other alternative strategies to increasing the amount of a drug that might get into the blood-brain barrier. So the screen that I talked to you today about was focused on the compounds which themselves can get through, but another part of the project is also seeing if there are drugs which by themselves don't get through the blood-brain barrier, but they have a fantastic rescue effect on the mitochondria and the lysosomes, and therefore could we help them to get through the blood-brain barrier. So we are in parallel doing some of that research. I just didn't have time to talk about it today.
Thank you. Back to Miratul from Richard Windle. Is it likely that there are other genetic defects linked to Parkinson's, or do we think we've found them all?
No, that's a very good question. No, there is this missing, there clearly are patients who have unidentified mutations in genes that are not yet known. I think the bottleneck now is that for geneticists, this is so the geneticists have really, I guess, they've solved all the low-hanging fruit. So if you have very big families with many people affected, that's a geneticist's dream. So a lot of those kind of families have already been, solved as to what their cause is. But now you have, individual cases in a family which are much harder because all of us carry, probably, all of us carry 30 to 40 different genes that are very significantly, mutated, but it's not clear how those affect you. So you can't just sequence someone's gene and say, genome and say, oh, this must be causing your disease, because there's all these other variants that you carry. And so I think that I can't tell you exactly how many more genes are waiting to be discovered, but I certainly think that in the next few years we will hear about more genes. I mean, it'd be great, for example, all the kind of work that we do, we try and understand signaling pathways, and using other approaches, we try and identify sort of control mechanisms for these pathways. And one of the things that we do, and my colleague as well, Dario, does this, if we find control pathway components, we always ask the geneticist, is this something that has come up in your patient screens? Because that's another way potentially of narrowing it down. But no, it's good. No, I think we'll have more genes in the future.
Thank you. This one's for Heather and Lara from John Telford. You included, oh, thank you, John, oligodendrocytes as target cells. These are at the heart of MSA, an atypical Parkinsonism. How far can your work be applied to MSA, and are you actively doing so? Atypical Parkinsonisms are an important remit for Parkinson's UK, and it is a devastating disease.
Yeah, so in terms of looking at oligodendrocytes, we have the tools to do that. We are not doing it at the moment. I take the point that definitely it is another important cell type that is involved in several variants, and we do know that they are involved heavily in several neurodegenerative diseases. We just haven't started with oligodendrocytes yet. We decided to start looking at astrocytes, so it's just kind of like a matter of time.
This one's from Keith for David and or Heather. Why do drugs aimed at cancer and diabetes, etc., have an effect on Parkinson's, and is there any underlying theory?
Yes, another good question there. Sort of diabetes and Parkinson's do sort of overlap to some degree, that there is like an energy starvation in both disorders. So this is why some of the drugs which have been shown to be effective in treating diabetes have actually been trialed in Parkinson's as well. So there seems to be an energy imbalance in both disorders. So there is that commonality there.
Right, thank you. I've just got three further questions that are around information that we could be sharing. So what we're going to do is, over lunch, we're preparing a slide that we're going to put up. So one question is, how do we find Citran on Twitter? That will be up there. Martin Taylor, what's the name of your Facebook page? We're going to put that up as well. That's an online Facebook research interest group medium, which you might be interested in. And, oh no, that's the one about the printed material. So yes, there will be interesting websites and information up over lunch. So I'll let you all go now. Thank you very much, and thank you to our presenters. Thank you very much.
Thanks very much, Judith and Gaia. I don't know about the rest of you, but this time of day I normally have a nice kip. But that looks so beautiful. I think in future I'm going to be. Okay, it gives me pleasure now to introduce John Sheepy and Professor Stephen Smith, who are going to make some presentations for you.
Now's the time to have your kip. I apologise for having to use notes, but I've got Parkinson's and find it very difficult to remember things nowadays. Good afternoon, my name is John Sheepy. I'm an oh, sorry, that's the wrong speech. As I said, I have a condition called Parkinson's. Also, I have a serious heart condition and spent the whole of August in the LGI at Leeds. I had expected to shake it off when I was given Parkinson's, but it hasn't worked out that way. It's quite sad memories brought back to me today because I don't come to Birmingham very often. In the 70s and 80s, I was a policeman. And the last time I was in Birmingham city centre and a night was on duty the night the bombs went off, so it's been quite moving. I went for my flu jab the other day, and the nurse said, oh, I've got to go and fetch something, and so I had to look at my notes while she was there. Have you ever done that? The doctor heads it as, this is a very strange man. Many people, my doctor, my neurologist, my Parkinson nurse, my heart failure nurse, and indeed my family say I'm strange. The reason for that is I don't let things get me down, not even this bloody Parkinson's. I won't let it win. So why am I here? As many times people have said that. Why is research important to me? I'm one of 9 children. Born in Warwickshire. My parents weren't Catholic, they were just careless. 1983 saw my mother's 62nd birthday. Then out of the blue, she calls all 9 of us together and we go round to a house. We think, God, she can't be pregnant again. No, she had shaking hands, dragging feet, Loss of sense of smell, loss of dexterity. Sound familiar? She'd been diagnosed with Parkinson's. Many of the family felt sorry for her. I gave her a job application for a cocktail shaker. Sadly, she died in 69 of a stroke. Roll forward to 2009. I'm 62. Yes, I know I don't look it now. I noticed shaking hands, dragging feet, loss of sense of smell, which is a good thing, so I know I only have to bath once a year. Loss of dexterity. You got it, I was diagnosed with Parkinson's. But I wondered back to my police career, whether I'd got it then, because at the age of 23, at our passing out parade, 93 policemen, and I was the only one in step. It takes thinking about. In 2007, I would hate it if my daughter, see, I'm not so careless. I'd hate it if she was diagnosed with Parkinson's. I think as a result, we have a duty to find a cure now. I am actively involved with the Parkinson Research Network, inspired by the amazing Anna and her team. They're absolutely brilliant. I feel it's our duty to work now towards a cure. At the same time, in Harrogate branch, we've encouraged a closer network tie with the doctors there, Dr. Buccalario. Things are going well. She's starting to involve us in everyday items regarding Parkinson. But don't get me wrong, it's a two-way. We put ourselves out for them. For example, she rang me one day and asked, could I go and give a talk at her daughter's school on growing old? Look at me, I'm not old. Oh, I don't know about that, but off I went, and on arrival she said, don't look so eager, walk slower, shake a bit. I get to the school and I'm introduced as Mr. Sheepy is a friend of mine. He's here to show you the problems when you get old. Through my involvement, I've had found some amazing people who dedicate their talents to helping us. Dr. Bannerman and Heather Mortiboys from Citra and Sheffield, Chris Elliott and Stephen Smith from York University. All are willing to help attend branches and help spread the word. They're fantastic speakers. I introduced Dr. Smith because research is very interesting. You see, the fire alarm goes off. I'm sure you will enjoy his amazing work, and I know through the encouragement I've given that Harrogate Hospital are trialling what he's going to talk to you about, and I hope you find it half as amazing as I do. I give you Dr. Smith. Got to let him see what he's reading.
Okay, well, first of all, thank you very much, John, and thank you all for giving me the opportunity to tell you what we're doing at the University of York, Department of Electronic Engineering. I'm not medically trained, I'm a computer scientist by training myself, but what we're trying to do to help people with Parkinson's, and more importantly, with the help of people like John and the Parkinson's UK local branches, try and get some of this technology actually in use.
Didn't touch anything.
I've got the right button now. So what I'd actually like to tell you about is two devices that we're currently developing. The first one is for monitoring the side effects of medication for Parkinson's. So I'm sure as many people here are aware that although the medication is really good, over time some people have problems with side effects that can lead to involuntary movements, and these can be quite severe, and they can certainly affect the quality of life, not only for the person with Parkinson's but also their carers. It's also clear from data that these can lead to falls, and falls are probably the most expensive aspect of caring for people with Parkinson's. These side effects are commonly known as dyskinesia, and these side effects can be managed, but only if the specialists have sufficient information about how frequently and in what degree these side effects occur during the day compared to when the medication's been taken. Now, many people with Parkinson's have quite complex medication regimes, and so really understanding the link between particular medication and these side effects is a really complex affair. So what we've been developing is quite a straightforward solution. So in the idea, the solution is that we need to monitor people with Parkinson's over a 24-hour period, so we can really get a very clear idea of when these side effects, these involuntary movements occur, how severely they occur, and then we can link them to the medication that's being taken. To do this, we use some simple sensors, and these sensors, you can see they're a picture of one, and we've got some outside, so you can have a look and see what they're like. We buy these in, they're nothing special. They have electronics in them, like accelerometers and gyroscopes. These are sensors that you'll find in most smartphones. And they measure the movement of the person in their own home over a period of 24 hours. They're easy to use, they don't hurt, they're non-invasive, and they're pretty tough as well. The way that we've trialled this with our partners, our clinical collaborators in Leeds, is that a nurse specialist will go and visit the person in their own home, so there's no need for them to come into clinical hospital. We show the person how to attach the sensors, and they just use simple Velcro straps. There's no need to switch them on or off, they're already left on, and then the person can go about their normal daily lives for the next 24 hours. And you can see we attach these to the biceps, the thighs, and the chest, and the head. Now, it doesn't have to be 6, but that's a clinical decision. It's up to 6 of these sensors that are used, and I can tell that the first reaction they have is, oh, this doesn't look very comfortable, but certainly through the help that we've had by working with people with Parkinson's, they assure us that they're certainly not too bothersome, and certainly it's only a period for 24 hours. It's also perfectly acceptable to take them off to have a shower, or if they're uncomfortable. And although we won't get measurements for that period, it's really getting a measurement over the 24 hours that's important. And so once that 24 hours is over, then the sensors are returned to the hospital. Either a nurse will come and pick them up and just check the medication regime, or they could be put in a jiffy bag and sent back by courier. But once these sensors are back at the hospital, they're then placed into a dock here. So those are the 6 sensors. They're just placed into a dock. That's connected to a PC, and we upload all of that data. And then this is where the really smart bit happens, and this is where we look at all those movements over the 24 hours. It could be, making a cup of tea, it could be out in the garden, it could be just sitting doing a crossword. But the software algorithms that we've developed are able to discriminate between those movements which indicate dyskinesia, this side effect of too much medication, and discriminate that from any other normal activity that the person has undertaken in that 24-hour period. And to do this, we use a type of computer algorithm called evolutionary algorithms. And they're called evolutionary algorithms because they're inspired by Darwinian evolution. Now, what on earth does that mean? Well, just very simply, most computer programs are written line by line, and they're written by a programmer to tell the computer exactly what to do. Computers are pretty dumb, but they do dumb things really quickly. With these evolutionary algorithms, we don't tell these computer algorithms what to do. We have a population of them, as Darwin would say, and they learn the difference between these movements. They learn the difference between those movements associated with the side effects of the medication and discriminate from any other movement you might happen to do. And so over time, we work these algorithms and we evolve a very good, fittest algorithm that will do this very well. Anyway, back to the data. So once we process this data from the person we've measured for 24 hours, we then automatically generate a time chart. Now, this was developed in close collaboration with our clinical partners. And very simply, what it shows is a time across the 24 hours. So in this case, we started at 3 o'clock in the afternoon or just shortly afterwards. And we can see there some dots and some lines. And I'll show you more closely what these represent in just a moment. So we have really a record of when the side effects in the medication has occurred, as these green lines, and the red dots are when the medication was taken. And so we know which medication was taken. We got a table down here which lists the time the medication was taken, what that medication was, and what the dose was as well. So if we have a quick zoom in, so we're zooming in to make it easier to see here, just a part of that time chart. We can see here at 9 o'clock in the evening, we had this drug was taken, 100 milligrams, Stalevo, and the clear lines where white means nothing's happening, but the light green lines are an indication that the algorithm has detected the side effects of the medication, this dyskinesia. And the light green means moderate dyskinesia, and the dark green lines, which I hope you can make out there, are severe dyskinesia. And these actually relate to the clinical scales used in hospitals. And then finally, this purple line at the top there indicates when the patient was asleep or went to bed. And so overall, this chart is very important, very useful, because now the specialists can see when the dyskinesia is occurring and relate it to when the medication doses were taken. And armed with that information, the specialists now have a far better idea of which medication to adjust and by which degree. So that's the first device. The second device I just want to spend a couple of minutes explaining to you is how we can improve and confirm diagnosis of Parkinson's and also track the development of Parkinson's over time. I think it's widely agreed that in the early months of somebody presenting symptoms, it can be very difficult to arrive at a confirmed diagnosis because these symptoms can be often confused with other Parkinsonian type conditions. So this device is based on the standard finger tapping test, and I should imagine most people here are familiar that one of the tests that they do in hospitals is in clinics, they'll ask you to tap your finger and thumb as quickly and as widely as possible. And this is evaluated by a skilled specialist, and depending on how well you can do that, they can learn something from that activity. Now, what we're aware of is that there are lots of really invisible or hard to see hesitations in the way in which the fingers tap. And so we attach some sensors which are able to track the movement of the finger and thumb in real time. And so we're digitizing that movement and feeding it into the computer. And these graphs indicate what's going on. So this first graph here, this is showing a finger and thumb opening. So this is the separation between the finger and thumb, and this is over time. So as the finger and thumb open, we can see the separation increase, and then as it closes, it comes down again. As we can see here with the person with Parkinson's, it's very similar. We get quite a smooth opening and closing again. But if we now look at the acceleration of that movement, the little kick to open the finger and thumb, for somebody without Parkinson's, we see a little acceleration as it opens, and then a little deceleration as we're at the top, then quite a strong kick to close it, and a very strong deceleration as they snap close. However, if we look at the same movement with somebody with Parkinson's, we can see it's very jagged. It's not smooth opening and closing, but it's actually quite a change in acceleration and deceleration as that occurs. So this type of technology is able to show us those imperceptible hesitations and stutterings that we hypothesize are indicative of bradykinesia, one of the symptoms of Parkinson's. And just finally, these grey lines here, these are the areas of these graphs that those evolutionary algorithms I described earlier had evolved to choose those parts of the movement of the finger tapping to make its decision on whether somebody had Parkinson's or not. And I suppose that's not a surprise really, because it's looking at the opening phase and then the closing phase of that finger tapping movement. So that's all really great. So we developed this technology, but how can we get it into use? How can we help people with Parkinson's benefit from this work that we're doing at the University of York? Well, there are two things. One is that I've been very grateful to the Royal Academy of Engineering. They gave me some funding to buy me out from teaching to help set up a spin-out company called Clear Sky Medical Diagnostics, which is spending time trying to commercialize this technology. But even that isn't enough. What we really need is the help of specialists, specialist consultants who know and understand what they're looking for, the specialist nurses who deal with the people with Parkinson's and really understand what it's like. But now I understand we also need to speak to people with Parkinson's through the local branches to see what they think about it. Because if we don't get their opinions, then we're not going to really develop anything that's successful. And so here's some pictures of John and his members of the local branch at Harrogate, and they've been absolutely superb in being able to identify and help us with the straps to attach those first sensors onto the limbs. Now you might think the straps are an easy thing, but of course it's one of the biggest problems. They have to be comfortable, they have to be disposable to satisfy the infection control, and there are many other aspects about making them as comfortable as possible to wear for 24 hours. So John's been great in helping get help from the Harrogate branch for that. But then really the way forward is how can we also get them introduced and trialled out in different hospitals. So this is one time I was in my office and I got this call, and it was John. And I'm quite used to the occasional phone call from John, but this one he says, my consultant Dr. B's in her office at her desk now, ring her right now. So slightly nervously I rang her and she said, oh, John's just spoken to you, hasn't he? And so she agreed for me to go and speak to the people at Harrogate Hospital to explain what we're doing, and now as a result of that we have some of the equipment in Harrogate being used as well. And so really just to finally say that as with all this type of work, there are many people who have helped along this journey, the people from the University of York and my other overseas collaborators as well, but also the Parkinson's UK branches, certainly in Harrogate, but also in Scarborough and Ryedale as well. They've always been fantastic, keep on inviting me back, interested in knowing how things are going, and very encouraging. So thank you very much for your attention. If anybody wants to see the devices, we have them outside and can look at them over coffee. Thank you.
Okay, thank you very much indeed. To John and Stephen, I just said to John, he described himself as a very strange man. I actually think he's a very funny man. Made me laugh. So thank you very much indeed. Dale to the stage, who's going to make a presentation to you. Thank you.
Hello, everybody. Oh, good. I'm definitely on, which is brilliant. For those of you who don't know me, my name's Claire Bale, and I'm the Head of Research Communications and Engagement at Parkinson's UK, but I think most of you do know me by now. And I'm the last person you have to listen to today. You've made it this far. I'll try and make it as painless as possible. And it's my job to really give you a bit of a sense of what we've achieved in 2017 in terms of research, but also in terms of the RSN, because the RSN has had, I would say, a very bumpy year this year. So I'm going to try and pick out some of the highlights from the research this year, and then also talk a little bit about how the RSN is supporting the whole research field to move forward. And we've heard some fantastic stories of that already today. Okay, so firstly, I think it's really important to give you an update on where we're at with actually developing better treatments and hopefully a cure as fast as possible. That at Parkinson's UK is our mission. And to do it, we have to make this work. So we have to make sure that we're making scientific discoveries, understanding and unraveling the secrets of Parkinson's and the mysteries that will one day lead us to a cure for the condition. Things like Miratul's work to understand the genetic basis of Parkinson's and what's actually going on inside our brains and our brain cells. Then those discoveries need to be taken forward. They need to be taken forward and developed into tangible treatments that can actually make a difference, that fix the problems. And that bit, as we heard from David, is actually one of the bits that hasn't always happened as well as it should in the past. And then if we manage to develop a new and better treatment, we need to be able to test it really effectively and efficiently in clinical trials in people with Parkinson's. We need to be able to see the benefits and measure the benefits if we're going to get those treatments eventually approved by regulatory authorities who rubber stamp them and say, yes, this is a treatment that works, it's safe, it's effective, and it should be made available to everyone with Parkinson's. So this is a really big challenge, and we are doing as much as we can across this entire pipeline to make things faster and more efficient. So we continue to fund excellent science at universities and hospitals around the UK, and we've heard about some of those projects today, which are finding out what's going wrong in Parkinson's, developing new ideas for how we can potentially treat Parkinson's better, both in terms of drugs and therapies, and things that are about improving everyday life with the condition. And we've introduced new things. David mentioned our Parkinson's virtual biotech, which really got up and running this year, where we're not just sitting back and hoping that someone else will pick up these fantastic discoveries and progress them for us. We're actually taking the initiative, we're looking for those opportunities, and we're proactively making the best ones happen. And I'll mention the first project in a bit more detail in a moment. And our biotech ultimately aims to have projects that take the very early stage discoveries and develop them into new treatments, but also to ensure we can run clinical trials of the most promising new therapies to get them further down the pipeline. And then finally, the Critical Path for Parkinson's is our global international initiative where we're saying we're working with researchers, we're working with companies. 9 pharmaceutical companies have joined this initiative, and we're working with regulatory authorities and other charities like the Michael J. Fox Foundation and Cure Parkinson's Trust to make clinical trials better. Because when we get to that stage where we think we've got something that really could work for people with Parkinson's and we test it in people for the first time, we need to be able to be confident that we're testing it in the best way we can, and we've got the best chance that it will be successful and that we'll get a result that means something. And too often, clinical trials give us results that we need to do more trials to understand better. We need to be more efficient, and that's what the Critical Path for Parkinson's is aiming to do. And we're actually holding a lecture in November in London where we're going to be delving into the critical path for Parkinson's in a lot more depth. And we will hopefully also be live streaming that lecture. So we will definitely be sharing that information with you nearer the time. So if you can, do come along online and watch that lecture. So this year has really been, I mean, we're just over halfway through our research strategic period, and we had huge ambitions, and this was the year, I think, that it all really came together. Our Parkinson's virtual biotech became a real thing, and the Critical Path for Parkinson's as well is really up and running. We hope we will have the first real success from the Critical Path for Parkinson's before the end of the year to tell you about. And also, this year, we are on track to invest the most we've ever invested in a calendar year in Parkinson's research. We're on track to invest more than £8 million this year, which is absolutely phenomenal and fantastic progress. So, what are my highlights, but also hopefully your highlights from this year? I've just picked 3, which was very hard, but I'm going to start with our virtual biotech. So, this is the idea that we're actually in the driving seat now. We're taking the best ideas that are coming out of universities and hospitals and taking them forward to develop them into new treatments for Parkinson's. So our first project was launched in March this year. And actually, it was a project that came out of the Sheffield Institute for Translational Neuroscience, which you've heard a lot about already today. And a project that actually was initially about finding new treatments for motor neuron disease. And this just goes to show again that there's so many connections between these different kinds of neurodegenerative conditions, and so much we can learn by collaborating across different conditions and disease areas. So Dr. Richard Mead, who's based up there, had this great idea and had identified some potential new drugs that could be really helpful for protecting brain cells from things like inflammation and oxidative stress, processes that we know are involved in the struggles and death of brain cells in Parkinson's. Now, we've worked together with the University of Sheffield, with Richard Mead, to set up a virtual biotech company, a spin-out company, that can take these exciting, promising molecules forward to actually turn them into drugs. And that work is being done by a specialist chemistry company based in Nottingham. And the project has now been going for over 6 months, and we're managing it in-house. So you may have met Jan today. He was here. I think he had to go early, but he's our drug discovery manager. And it's his job to work closely with the team and manage the projects within our virtual biotech to make sure they stay on track and they hit their milestones, because we only want to be investing in these kinds of projects if they're delivering real benefits. If they fail, we need to be able to cut them off and choose something else to invest in. But luckily, this project is delivering. So it's 6-month milestone. We said, yep, it's going well, and we can continue to invest and take this forward. And Richard was kind enough to do one of our lectures earlier this year, so we'll be making a video about the project available, hopefully very soon. But ultimately, if it's successful, we hope to have drugs that we can take forward into further development that will one day, if successful, be able to slow, stop, or reverse Parkinson's. So this is a very innovative way for a charity to be working. We're one of the only charities doing anything like this. And it's been fantastic since we launched this first project, that lots of other scientists and companies have been coming towards us and saying, we've got great ideas as well. Do you want to work with us on this molecule or this idea? And we've had lots of things coming in, and as David mentioned, we've now identified our second project, which is quite a different project, much further along, so much closer to being ready to test in patients. And we hope to be able to share more information about that before the end of the year, and even possibly launch our third project within the virtual biotech before the end of the year as well. So this is really taking off, and it's making a big noise, actually, in the charity sphere. Lots of other charities are saying, oh, how are you doing this? And we'd like to do it too. So it's fantastic and very exciting. So that's my first highlight of the year. My second highlight, although I was hoping to be able to tell you a lot more about it today, is that our GDNF trial, which I'm sure lots of you are familiar with, completed successfully earlier this year. And we are now impatiently waiting for the results. So for those of you who are not familiar, I'll just quickly explain what the GDNF trial is. So GDNF is a drug, but it's also something that we produce naturally inside our brains. GDNF is a chemical, a protein that our brain cells produce that helps to keep our brains healthy. It helps brain cells, especially those affected in Parkinson's, to survive and grow and connect with each other. So it's really, really important. And in the lab, when you give GDNF to brain cells, you can see them survive and grow and regenerate if they've become damaged. So the real hope with GDNF is that it could do the same in people with Parkinson's. And as you can see here, this is Christine, who's one of the participants in the study. GDNF is not a simple drug to give. You can't give it in a pill. So the trial that we've done, which has involved 41 people with Parkinson's, they all had to have surgery to have little tubes placed into their brains to allow GDNF to be pumped down the tubes to exactly the right place in the brain. And that's what Christine's having done here. So those tubes you can see pumping GDNF into her brain, which is quite amazing. And all of the participants, 41 of them, spent the first 9 months of the trial receiving either GDNF or placebo and not knowing which they were getting for 9 months. And then the second 9 months, everyone switched to receiving GDNF itself. So they knew they were definitely going to get the experimental treatment. And that process came to an end in February. And what was fantastic is every single person who started the study finished it. We had no one dropping out. We had no really serious problems with the treatment. So we are very positive about that. And the analysis now is pretty much complete. We are just waiting to find out when the results will be published and when we can share them with everyone. Some initial results were shared last summer, which showed just the very initial top-line findings from the study. That was a very limited analysis. They only looked at one measure at 9 months, so between the two groups, placebo and GDNF. And they actually didn't find a significant difference between the two, which was slightly disappointing. But we're not too disappointed because we know that actually there was so much other data that was collected throughout the study, so many other things that we needed to dig through to understand the effects of GDNF, that we're hopeful that the fuller results and the fuller analysis of all that data, and it really is a phenomenal amount of data that's been collected, will give us much more information about whether GDNF works, how it works, and whether it has potential as a future treatment for Parkinson's. And I'm really hopeful that we'll be able to share that information before the end of the year, or if not, shortly after the end of the year. And also, excitingly, throughout the course of this study, which really has been an incredible journey, especially for the participants, a BBC documentary has been being made following several of these participants as they went through this incredible commitment and going along to the centre and having brain surgery and going for every 4 weeks to have GDNF infusions. And the documentary itself, we expect to be 2 BBC 2 1-hour programs, and it will be shown sometime after the results are published, so hopefully not too long now. And I've seen some of the footage myself, and it is incredibly powerful, and we really hope that it will raise a lot of awareness about what Parkinson's is amongst those who don't know much about it, and also about the process of doing research and how clinical trials work. So do look out for that. We will definitely be sharing results with you, the Research Support Network, as soon as we possibly can. And please hold on a little bit longer because we are so nearly there. Okay, and number 3. So also this year, which you may have heard about, another important trial came to an end and reported results. And that was a repurposing trial. Now, we've heard a lot about repurposing already today, and this trial was a trial of a diabetes drug called exenatide, which I'm sure lots of you have heard about. In this study, 60 people with Parkinson's, with moderate Parkinson's, were involved, and it was a long-term study. People had either exenatide or a placebo, so a dummy version of exenatide, for 48 weeks. And then after 48 weeks, they stopped receiving any treatment, and 12 weeks later, they had their symptoms assessed all the way through, but they also had, so 3 months after they stopped treatment, they had assessments as well. And what was really promising was after this 12 weeks after they stopped exenatide, you could see a difference between the people who received exenatide and the people who received a placebo. It was a small difference, but people who received exenatide were very slightly better than when they started the study on a particular measure, which was their movement symptoms when they were assessed off their regular Parkinson's medication. And the people who received the placebo were very slightly worse than they were at the start of the study. So you could see that perhaps what was happening was exenatide was slowing or stopping the deterioration in Parkinson's, and the people who did not receive exenatide were continuing to slightly worsen as the condition progressed. Now, these results hold a lot of promise because they're some of the first that we've seen that suggest it may be possible to slow, stop, or potentially, maybe not reverse in this case, Parkinson's. But because this was quite a short study in some ways, 12 months isn't that long for people with Parkinson's in terms of deterioration, the overall difference was still fairly small. And we would expect potentially, if what we hope is happening really is happening, if you followed those groups for longer, you would see a bigger gap starting to develop as people are held and other people get worse. Some things, there were lots of question marks though as well. So although there was this difference, important difference in movement symptoms when measured off medication, there wasn't a significant difference in things like thinking and memory and quality of life that we would have hoped to see. But perhaps, again, if we did a larger study, a longer study, we would start to see that those benefits are there. So this was very promising, one of the first times we've seen a treatment able to do this in Parkinson's. But because it's still a relatively small study, a relatively short study, the next step is to do a phase 3 trial. And that is already being planned, and it will be for many more patients will be involved, and it will be for a longer period of time. And we're in touch with the research team, so as soon as we know more about that, we will certainly be sharing it with the Research Support Network, especially the opportunity to participate. So do keep your eyes peeled for more on this. The other important thing to mention is that exenatide is not the only diabetes drug being tested in Parkinson's. There are also 2 other similar diabetes drugs being tested for Parkinson's now. Those trials are actually underway in France and in the US, and potentially one of those could be even better. So we're also keeping an eye on how those progress and what results they show. So definitely something to keep an eye on. And it's one of the first times, the repurposing ideas that we've talked about today are already coming to fruition, which is fantastic. But I'm sure there's many more to come. Okay, so those are my top 3 research stories. You may dispute them. You may have others that were your personal highlights, but maybe you could ask about those in the question session. So let's talk a bit about the RSN and what you've been doing this year, because it really has been an incredible year. So, getting connected. This is what the Research Support Network started in order to do. Connect people affected by Parkinson's who care about research with research that's happening across the UK and the world, and also with each other. And this year, we now have nearly 4,000 members, which is absolutely incredible. We've increased by 50% this year over what we were last year, so that's fantastic. We have over 300 volunteers, and I know many of you here today are volunteers and doing many things for us, which is fantastic. We now have 6 local interest groups across the UK. So these are groups that work with researchers in their local area to raise awareness of research and also work together to develop ideas for research. And we also launched our blog, which I hope lots of you have found. So this is a blog that Becky at the back there really made happen, which we have on Medium, and we have articles on all sorts of different things. We highlight it in the roundup, and we've been working with volunteers to shape the content for the blog and make sure we're talking about the things that really matter to you. So do check it out. So yeah, we're more connected than ever, and we really want to continue to grow the network because there's so many opportunities to do things, and the more volunteers and people power we have, the better we'll do. Taking part in research. So far in 2017, 43 research teams have worked with us to raise awareness of their studies and given people in the network the opportunity to take part. So you really are powering research through your participation. And we launched a new thing on our website where you can actually put your postcode in and find the research studies that are closest to you. Because we had a lot of feedback from people saying, I really want to be able to see what's happening in my local area and be able to find those opportunities more easily. So we've created this thing on the website, and you can even filter those opportunities to find things that are more suitable for you. So if you only want to take part in things you can do at home, you can select for those. If you only want to take part in trials that are looking at new treatments, you can select for those. So we launched this really recently, and we're planning to send an email to the network soon to ask for your feedback. So please do have a look, have a play with it. Tell us what you like about it. Tell us how we can make it better, because this is for you, so you can find things to take part in more easily. And then finally, having your say in research. Oh, look who it is, it's Heather. She appears on most of my slides usually. It's amazing, it's the last slide. So this is really important as well, a really growing part of our work. We want to bring people affected by Parkinson's and researchers together to work in partnership so that you are actually having a direct impact on what research is done and how it's done. You're helping researchers to identify the questions they ask, make sure they're really relevant to people with Parkinson's, and then also helping them design studies so they meet the needs with people with Parkinson's. So if you're taking part in a trial, it needs to be developed to suit your needs, not just what's convenient for the research team. So you don't have to get up at the crack of dawn to go and have your brain scanned, for example. So you're improving research, you're making it better, more relevant, and more accessible to people with Parkinson's. You're inspiring scientists, you're giving them ideas, you're making them care more about the condition and want to do more research, and you're helping researchers understand the condition better. And we now have over 100 volunteers who've gone through our patient and public involvement training and are working in partnership with researchers in this way, which is absolutely phenomenal. So you've definitely had a busy year, and we hope that this is going to continue to grow and grow. And I think I'm probably bang on time here, so that's me done. Thank you so much, everyone, and for all of your contributions throughout this year, and yeah, for coming along today and participating. It's been fantastic, so thank you very much.
Okay, thanks very much, Claire. That was lovely. Right, if anybody has any questions about Claire's talk, can you please write them out on the cards and raise your hands, and then the Parkinson staff in the blue t-shirts will collect them. And I should just mention that any questions that are not answered by the panel this afternoon will be answered later on by email. Obviously, we haven't got enough time for everybody's questions to be answered, both from within this room and from the live screening. And the full question and answer list will be published on the Parkinson's UK website. Okay. Could we have the speakers from this afternoon's session back on the stage, please?
Love how everyone's really tired.
Okay, is that everybody? Right, let's start with a question from Laurel Miller for Judith and Gaia. The movements shown this afternoon are quite small. Other evidence from Australia, for example, suggests that big movements are key to maintaining fitness and reducing rigidity. One of the systems I know that she's referring to is called PD Power. Why are your approaches so different?
So I think there are different approaches can be used possibly in a complementary way, and they may be targeting different things. So the exaggerated movements, the big movements, might be good for maintaining those larger movements, those more general movements. And what we're looking at with the gestures and movements that we were talking about earlier is how you can actually incorporate the smaller, the finer hand movements that people need that are quite important for everyday activities. So you could potentially incorporate elements of both into one package that, you know, that has a broad range of effects.
Yeah, lovely, thank you. Okay, I've got a question now for David. Why does it take so long for existing drugs used for other conditions to be trialled for Parkinson's?
Yes, it's a question that I often ask as well. So the thing is that what we need to do is to make sure that the drug that is there for repositioning has a solid background to it, that it can be repositioned, and that it doesn't have any adverse side effects that may aggravate Parkinson's. For example, there was a drug that was mentioned in the literature earlier this year called trazodone, and that could be taken as a repositioned drug and taken into a clinical trial. But one of the potential side effects of trazodone is it could make people dizzy. So as you can see, that yes, it might change protein expression in the brain, but it might actually complicate Parkinson's. So we need to be very careful in our assessments about what drugs that we do take forward for repositioning. And obviously cost is another thing as well. You know, we can only run so many trials with our budgets, but if people want to run more marathons, you know, shake more tins outside of Waitrose and things, we can do more reposition trials as well. So it's a mixture of balancing the effects and benefits of potential drugs with actually managing what trials we can do with our budgets.
Okay, thank you. This question's for Stephen. I'm sorry, I don't know who it's from. Are the devices you're developing going to be used in clinical trials to help access the effectiveness of new treatments?
Yeah, and it's a very pertinent question. We certainly have in mind that they can be used for that purpose. So initially we're targeting at helping people with severe dyskinesia to see if we can help specialists find ways of managing their medication. But it's certainly true we are also interested in working with other collaborators, with even pharma companies to provide objective evidence of the effectiveness, efficacy of new drugs.
Okay, thank you. And I've got a question here from Andy Hobbs for Claire. Can DNA be a mandatory input into a clinical trial to allow more detailed analysis of outcomes? Given the diverse, unique way that Parkinson's presents in patients, is it realistic to expect a single therapeutic to work for everyone?
It's a really excellent question, and maybe other panel members want to chip in. So with GDNF, for example, they didn't initially take DNA samples to analyse as part of the results, but that's something they're doing now because they are interested in that, and I think it's becoming more and more a standard part of running a clinical trial, so that you can see, are there people with particular genetic backgrounds that particularly respond well or not well to a particular therapy. But it's hard, and actually, although genetics, we've heard a lot about genetics today, and it is really, really important, they're still quite rare, these genetic mutations. So, you know, even in a group of 41 people with Parkinson's, the likelihood of finding a number of people with a mutation is still quite low, even in younger people. So there's still a lot we don't know about the genetic background to the condition, which is holding us back, I think, a little in how we're able to analyse these things. But definitely, I think it is the future. I don't know if you want to add anything, David?
I think it's about the single targets, because if you think about it, there are lots of different pathways that we've already identified which might be leading to brain cells dying. And the thing is, at the moment, we're looking at drugs that often just inhibit one of them. So the other ones will be carrying on as they are, and just stopping one arm of these pathways might not be sufficient. And Virtual Biotech 3 project might tackle several of these mechanisms all at once. So rather than having one single magic bullet, we might be able to fire several different bullets at different pathways, and that might be a better approach for actually treating the disease.
Okay, thank you. Another question for Judith and Gaia. Are there any similarities between the dance performed today and Tai Chi?
We'll hand that one over to you, I think, the dance expert.
Well, some of the movements that I performed today are inspired from the Indian classical dance style of Bharatanatyam. So Bharatanatyam is movement, you have gross body movements and fine motor skills combined together. So we have a lot of hand gestures, or what's called as mudras. So through the course of my research, I identified that hand gestures can help improve fine motor skills. You acquire fine motor skills, new fine motor skills, and can you transfer those newly acquired fine motor skills to other activities that require the same? So I think this was one aspect that fascinated me and Jude, both of us. And so that's why the movements that we used today were inspired from Bharatanatyam, the Indian classical dance style.
Thank you. Another question for David from Graham Skinner. Loss of smell is a very early common symptom of PD. What are the reasons for this, and is this loss a useful prediction?
Yes, it's been known for quite a while that loss of smell occurs very early on in Parkinson's. And from your nose, your smell goes to what's called the olfactory bulb, which is in the basal part of the brain, and then it goes into the olfactory centers of the brain. And people have analyzed the olfactory bulbs within people affected by Parkinson's, and you can see cell loss within the olfactory bulbs as well. The problem is that the loss of smell particularly for different types of smells, also occurs in other disorders as well. Things like Alzheimer's, they also lose the capability of smelling different things. So what they're trying to do at the moment, some of the work is actually funded by Parkinson's UK, is doing scratch and sniff tests. So trying to find out which odours are particularly lost and whether you can actually distinguish like somebody with Parkinson's from somebody like we've got Alzheimer's. So obviously, if you can't distinguish the two populations, then it's very difficult to actually have a predictive test that might indicate that it's Parkinson's. But watch this space. You know, and one day we may have a scratch and sniff test to help with the diagnosis.
Okay, thank you. Question for Stephen. Are your devices available for people with Parkinson's, oblique stroke health professionals? If yes, how can they be accessed? If no, when might they be?
Yes, thank you. So we're just at the stage where LID monitor, that device for monitoring the side effects of medication, that has CE marking, which means that it has regulatory compliance. So it is available in the UK and throughout Europe for use. We're just at the stage where we're using these with a selected number of hospitals, just so we understand how the technology can be best used. So we are working with other hospitals, we are working through people with Parkinson's UK local branches to see how we can get these more widely available. But yes, we can make the equipment available, but we would like to work initially with hospitals and specialists so we understand how well it's being used.
Okay, thank you. Claire, this question's for you. Are the diabetes drugs being monitored for side effects if used by non-diabetic PD sufferers, for example, pancreatic damage, or at this stage are only diabetics taking part in the trials?
Yes, so part of any clinical trial that you participated in would monitor your side effects, any problems you experienced, whether they were related to the medication itself or other things going on in your life. I think in the diabetes drug exenatide trial, there were some side effects that they found, things like nausea, and actually one of the things that it can do is cause you to lose quite a lot of weight taking these diabetes drugs, and for some people that's great. For other people that can be a real big problem, especially if they're already a little underweight before they start. But no, the people taking part in the trials don't have to have diabetes. I think in fact they may even need to not have diabetes. So yes, it's really important and it is being looked at, but I don't remember anything about the pancreatic damage. But certainly they'll be looking for anything like that to make sure it really is safe.
Okay, thank you, Judith. Last question for you. If you don't have access to an exercise class, what type of exercise should people be doing?
I don't feel fully qualified to answer that question as I'm not a physiotherapist. But I think any type of exercise is considered to be beneficial. Obviously anything that can be done safely. Mm-hmm. As I said, we're looking at alternative routes, so if people are not able to go out to classes for whatever reason, there isn't something nearby or they don't have the mobility, there may be other ways, and people are developing lots of things now with virtual reality, augmented reality, or even things that you can already buy, like there's the Wii and the Kinect and things like that. So there are lots of options, you know, coming forward that people might be able to feel involved in an exercise program at home if they can't go to something physically.
Thank you, Jude. Okay, this question is for David and Claire. It is great that the research spend for 2017 is almost £8 million. What is the long-term strategy for research funding? Why aren't more of PUK's resources spent on research?
Do you want to go first?
Yes, okay. So in 2016, it was about 5.7 million. But by the end of the strategy period of 2019, it should be about 11.6 million. Now, as Steve Ford pointed out at the AGM yesterday, that as of the 1st of January, the piggy bank is empty. So for each year, we have to raise that amount of money each year to pay for the research spend. But don't forget, the money that is raised is not only spent on research. There's a lot of care support, there's the helpline, there's all the information services on the web that are very important for people with Parkinson's as well. So we need to have something that is for people with Parkinson's now, as well as for people with Parkinson's in the future.
Yeah, and the people who ultimately make the decisions about that balance and how the funding splits across the different strategic themes are the Board of Trustees, who are, you know, people affected by Parkinson's, people who know people with Parkinson's, and they'd have to make difficult decisions. But they are prioritising research in more than they ever have before, and that's because they're hearing how important it is to people affected by Parkinson's in the surveys that we do. And I think if that's what you think we should be doing, you know, by being part of the Research Support Network, you're showing how much you care about research. And you can always, you know, send emails, tell the trustees what you think they should do. That's what they're there to do, is to listen to everyone affected by Parkinson's and make the decisions based on the full needs of everyone.
I'll just say one point about the trustees as well. It was raised at the AGM that they would like more applications to become a trustee from people who are affected by Parkinson's. So then you can have your influence on the board, but they are lacking people wanting to come forward. And they do give you a lot of support with the role and things, but really it would be great if we have more people on the board. So if you want to consider registering to be a Board of Trustees, think about registering for next year's elections.
Okay, thank you. We're nearly done if you're looking forward to your afternoon kip. Stephen, there are many types of technology available today that could help people with Parkinson's. Would you suggest people invest in anything specifically?
So that's certainly true. There are many types of technology that have been developed for many years. In fact, I started looking into applying computers to helping people with Parkinson's way back in the 90s. And if you do a search online, you'll see lots of work has been done over this time. I think what's interesting is there's a big difference between doing some research and then being able to translate and commercialize that and making it available for people with Parkinson's to benefit from. And that's why we have the spinout company, and that's why we're working hard towards that end. So I can't give any specific advice on which ones I think are best. I think what is interesting that over the course of time, certain technologies will prove to have their place for different purposes. And it's really a case of seeing how well these do stand the test of time and how well adopted they are.
Thank you. Okay, Claire, quick one for you. What is a virtual company?
Oh, good question. A virtual company is a company that doesn't really have any staff, I think, or buildings or labs. So it's like a shell. And the reason they're so useful for things like our virtual biotech is that they can hold all the assets, so the intellectual property, and they provide a really simple way to make sure that, you know, the project is encapsulated in this virtual company and can be invested in by other people. It just makes it a lot cleaner than if we owned it or Sheffield owned it or we shared ownership. So our virtual biotech, we don't have labs. We're not building labs in 215 in London, and we have a limited amount of staff who work on the virtual biotech. We're really just being able to invest, and the work is delivered elsewhere. So in the case of the first project in the virtual biotech, that work is being delivered by a company that we contract to do the work for us, and they're a company that specialises in doing this kind of drug design work in Nottingham.
Okay, nearly done. One more question for David. 6 professional groups in Cambridge alone are working on Parkinson's, to add to Oxford, Manchester, etc. Do they communicate and collaborate? And that question's from Keith.
Yes, certainly communication is a big part of it. It's an important part. Certainly we have our own research conference which runs every 2 years, and that is a great opportunity for researchers working in Parkinson's to come together and communicate their results to each other. But as you know, a lot of the cohorts that Parkinson's UK fund, like Tracking Parkinson's, actually involves lots of centres anyway, so they are working together in a joined-up way, but we're always looking for different innovative ways in order to get people to work together a lot better.
Okay, thank you. Right, it gives me great pleasure to thank all the speakers today, and I just want to say something from my heart. We're so focused on, as patients, not knowing what causes our disease, and perhaps impatient because people haven't yet found a cure for us, that we forget to be very grateful for the researchers who are working to help us. And I thank you from the bottom of my heart for choosing to focus on this horrible illness which affects every aspect of our lives. It fills me with hope every time I come to a conference like this. Thank you very much indeed.
Thank you, Paula. I kind of wish that was the last word, but I'm going to say thank you and let you all go now. But before you do, in your packs today, there is a postcard, a Research Support Network postcard. What we'd really like each and every one of you to do is go away today and give that to one person you know who might benefit from being a part of the Research Support Network and help us grow and spread awareness of what we do. And thank you to all the speakers today and all of you as well. I think the takeaway message really is collaboration and innovation, and there's been a lot of evidence of that today. So thank you very much. Goodbye.